Hi everyone, it's Jamie Baxter, instructor for A&P2. We're going to continue on with our lecture on metabolism. Specifically, this part's going to cover protein digestion and protein metabolism. So in the last lecture, we were talking about lipids and how they were used in the process of beta oxidation to create ATP, lots and lots of ATP. And so let's talk about how proteins are used. So in the mouth, it's just mechanical processing that's occurring with mixing and churning. There's no chemical digestion in the mouth. It just begins in the stomach when pepsinogen is activated by HCl to form the enzyme pepsin. Pepsin will then begin digesting our proteins into smaller peptide and polypeptide chains. The strong acidity of the stomach is also going to help to denature some of those hydrogen bonds. In the duodenum, CCK is released to get our pancreatic enzymes to be released. They're always released as inactive proenzymes, and they will be activated when they're in the duodenum. So they're not activated when they're in the pancreas. So they're activated once they're secreted. Enteropeptidase is going to convert trypsinogen to trypsin. Trypsin is going to activate other pancreatic proenzymes into their active forms like chymotrypsin, carboxypeptidase, and elastase, all enzymes breaking down the tertiary and secondary structures of our proteins. So in the duodenum, these peptidases are going to be active in our intestinal lining. These are the like brush border enzymes that function in the intestinal lining to break down peptide bonds releasing amino acids. So dipeptidases break down dipeptides into single amino acids, right? Amino acids are the building blocks of protein. Just a quick review. So step 1, chewing breaks it down in smaller pieces. Step 2, turning and mixing the stomach. Activation of pepsinogen into pepsin, breaking down our proteins initially, as well as that acidic environment. In the duodenum, we have some of our enzymes breaking down our tri- and dipeptide structures and into their simple amino acid forms. So how are these proteins absorbed and transported? Small peptides and amino acids are absorbed directly into epithelial cells through facilitated diffusion and or co-transport. Because all amino acids are a little different charges, different sizes, different shapes, they're all going to act a little differently when coming into the cell. They're then released from the epithelial cells by the same transport mechanism that brought them in, and they're transported through the liver via the hepatic portal vein, just like carbohydrates were. So in the liver, again, just like it was kind of dipsticking and tasting the amount of glucose in the blood, it's going to do the same thing here with amino acids. It's going to adjust amino acid levels with more precision than glucose. So it can increase after less precise than how it controls glucose, because our blood glucose levels are much more closely monitored. The liver can actually use all of those amino acids to make plasma proteins like the albumins, the fibrinogens, and things like that are floating around in our blood, maintaining osmolarity. It can also create 3 carbon molecules from the amino acids in order to create glucose in the process of gluconeogenesis to create the pyruvate or acetyl-CoA for energy production. So epithelial cells are absorbing either through facilitated diffusion and or co-transport, bringing those amino acids in, getting them out and into the liver, where they are monitored and adjusted, either used to create our plasma proteins or 3 carbon compounds. So 10 of the 20 amino acids that we need can actually be made by our body. The other 10 are what we call essential, just like omega-3 and omega-6 fatty acids. We have essential amino acids as well, meaning they cannot be produced by our body. We have to ingest them in some way. And so therefore, maintaining a good diet is essential. Amino acids can be synthesized, other than the 10 that are essential. The creation of amino acids involves 2 main steps, or 2 main methods, I should say. Amination is a process where you're using an ammonium ion to form an amino group. An amino group is an NH4, which is basically an ammonium ion, and attaching it to a molecule. Transamination is the other process. This is where you're taking the amino group from one molecule and sticking it on another molecule to create an amino acid. So you're basically swapping amino groups. Here's the process of amination. You have alpha ketoglutarate, and you stick an ammonia ion on it. Notice that it is what am I going to say here? I had a word in my head, like a oxidation reaction where we're adding hydrogen, and in it we're creating that NH2, which is our amine group, sticking another hydrogen on so our carbon's nice and happy with 4 bonds, creating a water in the process. This is called a dehydration synthesis reaction where we're actually removing a water molecule to create a larger molecule. In transamination, we're literally taking a molecule such as glutamic acid, taking the amino group from it, and sticking it on an organic acid. Notice in this case, this double bonded oxygen, this carboxyl group right here, is basically swapped for the amino group. So you end up with a different organic acid and tyrosine in this case. And what determines an amino acid is that functional group. In this case, it's the blue boxed area is determining which amino acid it is. So the catabolism of amino acids requires first removing that amino group. This is tricky because that amino group, when it is released, creates a very toxic chemical to our body. The removal of the amino group can be done through transamination or deamination, simply removing it, but it's going to release ammonium ions. Those ammonium ions are toxic to our body. So our liver absorbs those ammonium ions when protein catabolism is occurring. Those ammonium ions are floating in our blood, and those ammonium ions are converted into urea. It's called the urea cycle. The urea is then excreted in the urine. It's what gives our urine that nice, lovely yellow color. And here you see amino acid catabolism. You have deamination occurring where we're literally pulling that amino acid off, creating an organic acid, which can be converted into a 2 or 3 carbon compound and ammonium ion. That ammonium ion combining with carbon dioxide to form urea in the urea cycle. You can, actually, I'm going to stop the video here because the next section does not have to do with protein catabolism. So, have an awesome day.